Although metabolism is often thought about in terms of isolated pathways, it's really more like an interconnected net. And the intermediates of one pathway are intermediates of another pathway too. And some of these reactions in those pathways are going to be easy to reverse. And so you can kind of more easily siphon things off into one pathway versus another, but then, oops, you change your mind and now you go back. Other reactions, however, are not going to be easily reversible. These are often associated with the hydrolysis of ATP or a similar quote unquote high energy compound. And we'll talk more about this and why this is. But these reactions are going to serve as sort of go, no go points. They're going to be hard to reverse. They're going to commit you to a certain fate. And so we're going to need to regulate them highly. We can regulate where molecules go throughout this network by controlling both the concentrations of the various intermediates, as well as by controlling the enzymes that catalyze the reactions. And so we'll talk more about when we do what, how we can know if a reaction is metabolically reversible or irreversible, why this is inherently, and how cells are able to control the flow through this network. So let's start by thinking about reversible versus irreversible reactions. Now, everything is technically reversible, but just because it's technically possible doesn't mean it's going to happen. So if we want to think about thermodynamic favorability or the spontaneity of a reaction, and I hate the word spontaneity because it makes it seem like, poof, it just is always going to happen. But even for those quote unquote spontaneous reactions, you're often going to need some sort of catalyst, something like an enzyme that's going to lower the activation barrier and help you actually do the reaction. But enzymes can only make the reaction happen faster, they can't make the reaction favorable. The inherent favorability of a reaction comes down to entropy and enthalpy, and it can be reflected with this delta G naught, the standard free energy change. The standard free energy change is going to tell us if we start with standard conditions, so concentrations of reactants and products are 1 molar each, how much free energy would we release in order to reach equilibrium? If you have a negative delta G naught, that's saying that you would have a kind of inherently favorable reaction, you would be making more products, and so you would have a negative delta G. If you got a positive delta G, well, now you're inherently unfavorable in the forward direction. You're actually favorable in the reverse direction. And if it's zero, then neither way is favorable. But remember, this is saying to get to equilibrium, this much energy would be released. And so we start at standard conditions where we have 1 molar of our products and 1 molar of our reactants. And so remember to take into account stoichiometric coefficients and things, which we're not going to worry about now. But we have these equal concentrations of our products and reactants. We plop them in and see, do we make more products, in which case we would have a negative delta G, or do we make more reactants, in which case we'd have a positive delta G. In both cases, this is going to be in order to reach equilibrium. But typically we're not at equilibrium because if we were at equilibrium, there would be no drive to go in either direction. So to think about the actual favorability of a reaction, we need to think about what is the actual ratio of products to reactants. So where in here are we actually plopping things? Because the further we are from equilibrium, the more energy is given off to reach that equilibrium. And if we want to give off a lot of energy, we're going to want to be far from equilibrium. So if we add more reactants, we're going to make the forward direction more favorable. And if we add more products, we're going to make the reverse direction more favorable. However, if we have a really large negative delta G naught, so it's going to be really, really inherently favorable in the forward direction, we'd have to really, really skew the concentration of products to reactants, where we have way, way, way more than products than reactants in order to actually get it to go backwards. And if we had a really large positive delta G naught, we'd have to have a lot more reactants than products in order to get it to go forward. That being said, even inherently favorable reactions, so ones with a really large negative delta G naught, are going to be influenced by the concentrations. And so even though we have some reactions that are so inherently thermodynamically favorable in one direction that we call them quote unquote irreversible, these are also going to depend on the concentrations. And so we'll see how even ATP hydrolysis, which is very, very thermodynamically favorable inherently, becomes more thermodynamically favorable if we increase the concentration of our reactants. Because if we were to let things go to equilibrium, what would happen is that we would just hydrolyze all of that ATP. If we look at a table, we can see that our standard free energy change is about minus 7.3 kilocalories per mole for ATP hydrolysis. This is going to be saying that if we were to let it go to equilibrium, we'd have between 100,000 and a million times more of the ADP of our product than of our ATP, our reactant. So we got to actually keep it from reaching equilibrium. It's really important that we do keep this reaction from getting to equilibrium because we can use these metabolically irreversible reactions in order to drive reactions that are thermodynamically unfavorable by coupling them to one another. And we'll get back to this concept of reaction coupling later in our lesson. Although life is driven by that desire to reach equilibrium, if you actually reached it, life would cease to exist. And so we keep equilibrium out of balance, but we still want to have kind of reliable levels of things. We want to have controlled levels of things. And this is going to introduce us to this concept called steady state. Steady state is where we're keeping constant levels of metabolic intermediates despite being away from equilibrium. It happens by forming, and this can be via multiple routes, and consuming, which again can be via multiple routes, or otherwise removing intermediates at equal rates, rather than forming and unforming them at equal rates. So unlike equilibrium or allowing the concentration of the reactants and the products to change, we don't have a closed system, but we can be keeping things away from equilibrium, adding reactants in, and then removing the products, but then adding reactants in from other sources and taking products to other sources. As long as the input equals the output, we're able to keep constant levels. If you think about a water pipe, if you close off the pipe, you can have a constant amount of water in the pipe. But you could also have a constant amount of water in the pipe if you flow water through the pipe, with the water coming in equaling the rate at which the water goes out. Similarly, we can have that sort of thing happen to keep concentrations of intermediates of pathways at specific levels that are away from equilibrium, so you still have a drive to move things forward. And note that I'm using oxaloacetate because oxaloacetate is a really great example of this. Oxaloacetate levels are kept really low, and they're kept low to make it so that this malic dehydrogenase reaction is favorable in this direction where you go from malate to oxaloacetate, even though it has a positive delta G naught. So let's step back and think about how we can know without knowing the delta G naught whether a reaction is metabolically reversible or irreversible just by looking at a diagram. If a reaction is drawn with double arrows between the intermediates or between the reactants and the products, that's typically going to indicate that a reaction is metabolically reversible. On the other hand, if you see a single arrow, that may or may not indicate that a reaction is metabolically irreversible, meaning that the delta G naught is too large to overcome under typical physiological conditions. So, for example, where I've drawn these arrows going both directions, that's indicating that a reaction is metabolically reversible. You can go back and forth more easily. Other arrows, however, are going to be drawn as single arrows. These single arrows are going to be things that are going to be metabolically irreversible. You can't just go back easily. And remember, those are going to be the points that we're going to want to really, really, really tightly regulate. Not all single arrows or single-headed arrows are going to indicate metabolic irreversibility. Sometimes it's just easier, and sometimes you don't know if it's reversible or not. So you might see these things with a single arrow that's actually metabolically reversible or vice versa. But if you see a combination of these double-headed arrows or these double arrows and a single arrow or a single-headed arrow, that's going to typically tell you that some of those are going to be metabolically reversible, the ones with the double-headed arrows or the double arrows, and then others are going to be metabolically irreversible, those with the single-headed arrow. And so in some diagrams too, so in my big roadmap, what I do is if the reaction is going to be metabolically reversible easily, I have the arrows kind of close to one another, and they'll have a single same enzyme associated with that arrow. Whereas if they are not easily reversible, it'll either have a single arrow or it'll have these two single arrows that are kind of separated and have different enzymes associated with them. If I have multiple arrows in a row too, this is going to indicate that multiple steps are involved. It's not just a single reaction. Typically what we'll have the enzyme name is going to be either above or below the arrow. And then you have curved signs that are going to indicate any sorts of additional molecules that are involved. Things like cofactors, things like your ATP, NAD, NADH, water coming and going, all these things. You might also see sometimes transient intermediates, and these are not reaction intermediates like we talked about with enzyme reactions. Instead, these are going to be actual molecules, actual kind of temporary intermediates. They're so temporary that they don't leave the site of the enzyme, and so sometimes you'll see those indicated in brackets. Also, the equations are often not balanced, especially for those protons. Now let's talk about why it matters whether something is metabolically reversible or irreversible, and how that dictates how flow is controlled through metabolic networks. Metabolically reversible reactions, but not the irreversible ones, can be controlled just by controlling the substrate and product concentrations, and we refer to this often as thermodynamic regulation. By controlling the amounts of the products and the reactants, remember those are intermediates of pathways, we can control the directionality of the reactions in those pathways. So a great example of this, again, is that malate dehydrogenase in the citric acid cycle, which converts malate to oxaloacetate. If we look at this reaction, we see that it's delta G not prime. So remember, the prime symbol is just indicating biochemistry standard conditions. It's actually going to be positive 29.7, but we're able to keep the delta G, so the actual free energy change, close to zero, because we are able to keep the concentrations of our oxaloacetate, our product in this direction, really low. So by keeping the oxaloacetate concentrations low, we're able to make it so that this reaction can actually happen in the forward direction. That is, to make oxaloacetate from malate. Because we're taking this oxaloacetate, we react it with our acetyl-CoA and take it back through the cycle. And so we're able to keep the concentration of oxaloacetate low in order to make it so that we've got a lot more of our reactants than our products. And so even though this reaction is inherently unfavorable in this direction, we're able to make it favorable, make it have a negative delta G, or at least a neutral-ish delta G, even though it has a positive delta G naught. So if we have these metabolically reversible reactions, we can see how just by controlling the concentrations, we're able to control the directionality. But then we have those metabolically irreversible reactions. Those reactions that are going to be hard to go back from. They're going to commit you to a pathway and or an end product. And so we want to really regulate them, but we can't just regulate them by controlling the concentrations. We'll see how it's still important to control the concentrations because that's going to, one, keep you from getting to equilibrium, and two, make it so that you still have a lot of oomph. But we also need to regulate them a little more directly. And the way that we're going to regulate them directly is by regulating the enzymes that catalyze them. So remember, the reason why I hate the word spontaneous is because it makes it sound like it's just like always going to happen like right away. But you're going to need a catalyst. Spontaneous actually tells us only about equilibrium. It tells us it's favorable to reach equilibrium in which the products are favored. It doesn't say that it's likely that that's actually going to happen. And so the enzymes are what's going to actually make it likely that it'll happen inside of cells, inside of organisms. So by regulating those enzymes, that is, we call this kinetic regulation, then we can control whether the reaction will happen even though we can't control which direction it would go if you just left it to its own devices without controlling those concentrations, because the enzyme is going to lower the activation barrier in both directions. So how do we regulate these enzymes? This can involve regulating the protein itself, so often via phosphorylation or dephosphorylation to activate or deactivate them. So we already saw that when we were talking about biosignaling, and we saw how we could have those phosphorylation cascades that were going to activate kinases that activated other kinases that activated other kinases. Similarly, we can activate and inactivate various enzymes in metabolic pathways. We can also have transcriptional regulation. So we also saw that another output of biosignaling pathways was often that you increase the transcription of various genes to make various protein products. And so that could regulate whether an enzyme is present. And then compartmentalization. So things like the citric acid cycle, those are happening in the mitochondria. But then there are also metabolic processes happening in the cytoplasm. And so we can make it so that certain enzymes are only in certain compartments, such as having certain enzymes only be in your mitochondria and other enzymes only being in your cytoplasm. And we could even do things like have isozymes, and we'll talk much more about this later, but different versions of the same enzyme in the cytoplasm and in the mitochondria that respond differently to different regulators. And so more on that later, but think about the kinetic regulation of these metabolically irreversible reactions. And then the thermodynamic regulation, regulating the concentrations in order to control the directionality of these metabolically reversible reactions. Another important one is allosteric regulation. And so there we have small molecules, often intermediates in that same pathway, that are going to be binding to the enzymes and affecting their activity. When we talk about regulating the enzymes, we're often going to see these symbols used. And so we'll see a lot of arrows. And so we already kind of got used to seeing some arrows from our biosignaling pathways. Activation, you're typically going to see things like green arrows. You might see triangles or plus signs. Inhibition, you'll see things like X's and minus signs, these kind of railroady looking signs. I don't know what you call these. And this kind of sideways T. Sometimes in red, these are indicating inhibition. What you will see often is two main types of regulation through these arrows. Not always, but they're commonly you're going to have pathways kind of regulate themselves to match supply and demand. And so you can have feed-forward stimulation where substrates upstream are going to be activating enzymes downstream. And so this can be through things like allosteric regulation, where you have a substrate bind to the enzyme and activate it. Or you can also have feedback inhibition, where you have downstream products that go and inhibit an upstream enzyme. And again, you're often going to see these happening at reactions that are going to be metabolically irreversible. You might have spotted a sort of problem. Metabolism encompasses both anabolism, making molecules, and catabolism, breaking down molecules. It's really important to remember that a negative delta G in one direction means a positive delta G in the other direction. Same magnitude, but different in sign. We need our pathways to have a negative delta G overall so that we're able to go down that pathway. Anabolic and catabolic pathways, therefore, cannot just be simple inverses of one another, because if the reaction pathway is favorable in one direction, then that pathway is going to be unfavorable in the other direction. Instead, we're going to have these pathways, so say glycolysis and gluconeogenesis, the breakdown of glucose and the making of glucose. They're going to share they're reversible reactions, they're going to share those enzymes that catalyze those. But then in the case of the irreversible reactions, they're going to use different enzymes. And not only are they going to use different enzymes, they're going to go through different reactions. Because remember that the enzymes cannot change the thermodynamic favorability. The enzymes cannot control that delta G. So we need to take those reactions where we have that really, really large delta G and take an alternative route. For example, in our glycolysis and our gluconeogenesis, we'll see that at these points where they have these metabolically irreversible reactions, they're going to be using different enzymes. And they're going to be going through different reactions. So take the first step of glycolysis, when we're phosphorylating the glucose to give us glucose 6-phosphate. This has the effect of trapping the glucose in the cell, and so we're committing the cell to keeping that glucose for itself. We've got this phosphate group. It can't go out through the glucose transporters. It can't just diffuse out, and so we're committed the cell to keeping this glucose. We haven't yet committed it to a certain fate. Instead, we're going to do that at these later steps that we're going to control. But for now, we've kept the glucose in the cell. We've done this by transferring a phosphoryl group from ATP onto glucose to give us glucose 6-phosphate and leaving us with ADP. And in this way, we've trapped this. And we were able to make this a favorable reaction by coupling the phosphorylation of glucose to the hydrolysis of the ATP. And again, more on this later. This had a minus delta G, which means that if we were to go and try to put that phosphoryl group back from glucose onto ADP to give us ATP, we would have a positive delta G. Instead, what we're going to do is we're just going to hydrolyze the phosphate group off of glucose 6-phosphate. So we take water and we attack it and we kick off the phosphate group, leaving us with glucose. And this is this glucose 6-phosphatase enzyme that only a couple tissues, so like your liver and your kidneys, have. And this is why your muscles can't ship out the glucose that they get from glycogen. They only get glucose 1-phosphate, which they convert to glucose 6-phosphate and take to glycolysis. But going back to this, both your muscles and your liver have this hexokinase that is able to phosphorylate the glucose and drop it into the cells. The reverse direction would not be favorable if you go directly to reverse this reaction. So instead, you use a different enzyme that does a different reaction to go in the reverse direction, this glucose 6-phosphatase. So not only are they different enzymes, but they're doing different reactions. This last step of glycolysis, where you're going from phosphoenol pyruvate to pyruvate, this step is actually going to require 2 enzymes and 2 different reactions and a whole additional intermediate in order to reverse it, because this step has a really, really large negative delta G. And so it's going to be really hard to reverse. We can't just change the concentrations of things without messing up everything going on in the cell. And so instead, we're just going to take this detour to go backwards. In this way, both glycolysis and gluconeogenesis are able to have negative delta Gs. But we'll see that with gluconeogenesis, we're actually going to be having to spend ATP and spend GTP, whereas with glycolysis, we're going to be making ATP. But by coupling the usage of the ATP and the GTP to the reactions that were metabolically irreversible in the other direction, we're able to make it so that we can make both directions, the pathways overall, thermodynamically favorable. As I mentioned, even inherently favorable reactions are going to depend on the concentrations. At an extreme, you can think about if you've got the ATP hydrolysis, which we said was thermodynamically favorable in the forward direction. If we had no reactants, well, we could only go from products to reactants. So it would be favorable in the reverse direction. And if we had no products We would only be able to go in the forward direction. And going in that forward direction would make it so that we get even more energy released than if we had started at a less extreme concentration skew. So by controlling the amount of the reactions in the products of those metabolically irreversible reactions, we're actually able to control the energy that they release. We're able to keep the energy currency, ATP, valuable. So let's talk about why ATP and similar compounds are thermodynamically favorable. Why are they valuable? Why can we use them as a source of kind of energy currency? So I say energy currency. I like to think about ATP as kind of arcade tokens. And so you go to an arcade and they have one of those token machines and you can stick in $1 bills, $5 bills, $20 bills if you're feeling really gambly. And it'll spit out a bunch of the same arcade tokens. And it doesn't care what denomination your bills were that went in. It doesn't even care where they came from. It just gives you the same tokens. And the same tokens can be used in any game. Similarly, we can break down all sorts of different molecules. All of these different molecules, we got fats, we got glucose, we've got amino acids. All of these can all be broken down, catabolized to give you ATP. And then that ATP can be used by various enzymes in your body. By coupling, it can be used to power unfavorable reactions. And so we've got this powerful token, this energy storage form. Doesn't care where it came from, but you can use these different fuel sources to make it, and then you could spend it in all sorts of different places. Like you can spend those arcade tokens in all sorts of different games. Remember that kinases are enzymes that add phosphoryl groups, and then phosphatases are enzymes that remove the phosphate groups. A couple other terms that you'll want to know are orthophosphate. This is going to be a form of inorganic or kind of free-floating phosphate group, as opposed to an organic phosphate group where we actually have it on an organic molecule. This orthophosphate is just a single phosphate group, so a phosphorus surrounded by 4 oxygens. You'll typically see it with like this pi symbol. And then pyrophosphate is where you have 2 of these phosphate groups, so you've got this ppi. ATP kind of stores energy in a couple of different ways. And so it's a common misconception that it's the breaking of that bond that gives you energy. The breaking of this bond does not give you energy. Instead, it's the hydrolysis of this bond that gives you the energy. The actual breaking of a bond is actually always going to be endergonic. It's going to take energy to break a bond. However, you have to think about what are you getting in return? So when we think about the hydrolysis of ATP, we're thinking about the reaction of the water with the ATP. And then we're thinking about the products where we have these ADP and this inorganic phosphate interacting with the water, interacting with other water molecules. Because this is happening in the system, we need to think about the state of the system before and after the reaction. In the case of our ATP hydrolysis, a few different things are happening. One is charge release. You have these 3 negatively charged phosphate groups held together, kind of like a clamped spring. So you've got a lot of repulsion between those negative charges. When you hydrolyze it, you're going to basically unclamp the spring. And if you were to, say, take a spring and act like an actual physical spring and have a clamp on it and then let go, the spring would kind of bounce out, and maybe you had a ping pong ball at the end and it would shoot the ping pong ball across the room. Similarly, we get this release of the charge repulsion when we hydrolyze that bond. And now we're left with this ADP and this orthophosphate that can go hang out with the water. And so you get these favorable hydration reactions. You also get better resonance stabilization of this inorganic phosphate, this orthophosphate, than you have in the form where you have these all connected. There are other things that happen, ionization of your ADP, various things like this. And the important thing I care about is that you know that it's not the breaking of a bond that generates the energy. It's the actual overall process of the ATP hydrolysis. And it's not just ATP that this is applies to. And so we have other phosphorylated compounds that actually have a higher energy of hydrolysis, standard free energy we have hydrolysis. Things like our phosphoenolpyruvate. This is what I was talking about, that last step in glycolysis, that we actually need two enzymes to go bypass, and we go through a different intermediate because that is so thermodynamically favorable to go in this direction of hydrolyzing the phosphoenolpyruvate. But rather than just hydrolyzing it, we're actually going to transfer that phosphate group onto ADP in order to give us ATP. Because the ATP bond wasn't actually the one that was going to give us the most bang for our buck. So remember we said that ATP hydrolysis was going to be about minus 7.3 kilocalories per mole. So we would have between 100,000 and a million times more of the ADP than ATP if we were at equilibrium. But remember that if we were at equilibrium, then we'd be dead. Life is driven by that desire for equilibrium. We need to keep it having that desire for equilibrium, because if we're at equilibrium, then our hydrolysis is not going to be useful at all. As our ATP levels diminish, not only does the amount of our fuel diminish, but the fuel itself is actually going to lose potency. Its hydrolysis is going to give us less energy than it would if we were further from equilibrium. So we need to keep high ATP to ADP ratios, and as we'll see, high ATP to AMP ratios in order to keep ATP as a good fuel source. And in fact, that's what we do. And so if you look at the actual concentrations of ATP, ADP, AMP, and inorganic phosphate in various types of cells, what you can see is that we've got more ATP than ADP. And in some cases, significantly more. In our red blood cells, for example, our erythrocytes, we have 5 times more ATP than we have ADP. Despite the fact that if we were to let that reach equilibrium, we'd have between 100,000 and a million times more ADP. Instead, we've got about 5 times more ATP. So we're able to keep things even more favorable because we are making it so that we're even further from equilibrium than we would be if we were to be at that standard conditions. Because remember, the delta G naught is telling us about the standard conditions where we had 1 molar of our products and of our reactants. So we're starting here. Instead, what we're going to have is we're going to have it so that we have a lot more of our reactants. We're all the way over here. We're further from equilibrium, and our equilibrium was already going to be further over here. And so we're just going to basically, the further we skew conditions to the left, the more we're going to make it so that the reaction is going to give us energy going to the right. Additionally, by controlling the concentrations of ATP and ADP and AMP, we're actually able to use these molecules as regulatory molecules, as signals of our fuel status. So if we've got more ATP, then that's signaling that we've got a bunch of energy. Let's go and actually use it. Let's make stuff. Let's do anabolism. But if we're running low on ATP, then we want to break down things to get more ATP, and we call that catabolism. So we'll get more into this later, but ATP is going to inhibit those catabolic pathways. We've got plenty of ATP. We don't want to break things down. We want to make things. And so we're going to activate the anabolic pathways. And not only do we want to activate the anabolic pathways, but we also need to be at the same time inhibiting the catabolic pathways, because we want to prevent ourselves from breaking down the stuff that we make. Conversely, if we have low amounts of ATP, that means we've got a lot of ADP and AMP. Those are going to actually activate those pathways that are going to produce more ATP, and they're going to inhibit the pathways that are going to use the ATP that we have. So we saw that ATP was helping control the levels of other things, but how do we control the levels of ATP itself? We're going to have to use favorable redox reactions to drive this production of ATP from ADP. Redox reactions in which we're passing the electrons from these fuel molecules onto these carriers, NAD+ and FAD, taking them to those electron transport chain, passing those electrons through more favorable redox reactions, pumping protons into the mitochondrial inner membrane space. Then we're making a gradient. We saw that gradients are going to drive things. We got this gradient of protons. This gradient of protons is going to drive the synthesis of ATP. So that's how we can make ATP in kind of a sustainable manner. But how do we keep steady concentration of ATP in a pinch? There is an enzyme called the adenylate kinase. Remember, a kinase is going to add phosphoryl groups. What it does is it takes two ADP molecules, joins them up, takes the phosphate group from one of the ADPs, puts it on the other ADP. That gives you ATP and leaves the other one as AMP. And so this is going to lower the ADP concentration and replenish the ATP concentration during periods of intense demand for ATP. And there's a similar enzyme, guanylate kinase, which will do the same for GTP. Note here that when we are doing this, we are making more ATP, but at the same time, we're also making AMP. And relatively speaking, we're increasing the AMP by a much higher percentage than we're decreasing the ATP. For example, if we were starting with 5 millimolar of ATP and we use 0.5 millimolar of it, that would be a 10% change in our ATP levels. But if we were then ended up with going from 0.1 millimolar to 0.6 millimolar of AMP, well, booyah, that was a 600% increase in the AMP. So our AMP is going to be a more sensitive indicator than ATP when it comes to regulating things. AMP is going to have a variety of different effects, and one of it is going to bind to AMP-activated protein kinase, or this AMPK. And then that AMPK is going to phosphorylate key proteins and regulate their activities in order to increase the production of ATP. Going back to ATP, ADP, and AMP being important for regulating the enzymes involved in metabolic pathways. And often they're going to be regulating those irreversible steps of metabolic pathways. But what if you don't have enough ATP and you need energy ASAP? Well, this is a situation for which your muscles aren't keen. So they stock up on energy stored in phosphocreatine. What happens is that in your muscles, you have this molecule creatine, and it's an amino acid, but it's not a proteinogenic one, so it's not one that we actually have in our proteins. But you can see it's got amino groups and it's got a carboxyl group. And what it does is it can actually go, and one of those amino groups can take a phosphoryl group from ATP to give you ADP and this molecule phosphocreatine. And then when ATP levels are low, it gets transferred back to ADP to make more ATP. And this is happening thanks to this creatine kinase. You still have to eat creatine because the creatine can get non-enzymatically converted into this molecule creatinine. And the reason why I'm mentioning this too is that your kidneys filter this out, and so you pee it out. But if your kidneys are having problems, this can alter the creatinine levels in your urine. And so doctors can test the creatinine levels in order to know that all is okay with your kidneys. But that's really mainly happening in your muscles, or in this case, the rat's muscles. So you can see that not in the liver, but you've got this creatine in the muscles that is then going to make it. So you've got phosphocreatine in the muscles, and that phosphocreatine in the muscles is there so that if you need ATP right away, you can transfer it to ADP, give you ATP, and run away from that bear. A few cool stats on ATP. If you have an intake of 2,500 calories, and remember those are actually kilocalories, that corresponds to the turnover of about 180 kilograms or 400 pounds of ATP. But your body only holds on to about 50 grams or a tenth of a pound of ATP at a time. And so in order to reach that 180 kilograms that you're using every day, it has to be constantly turning it over. So basically using that ATP. Each of the molecules of ATP has to get turned over or hydrolyzed 4,000 times per day. If you do the math, you'll see that that's about 6 times 10 to the 22nd ATP molecules that are actually being used by your body a day. So that's like 66 trillion molecules of ATP. And these are getting turned over, the last phosphate group, that gamma one, is getting taken off and added back on 3 times per minute. Pretty wild. And that's a really great book, by the way. ATP is often going to provide energy by group transfers rather than simple hydrolysis. So sometimes we'll see that we are just kind of hydrolyzing ATP, but other times we're going to be using the power of ATP in order to drive reactions. And all we're going to see is that above the arrow, we have ATP going to ADP plus PI. For example, the conversion of glutamate to glutamine. So the interconversion of these amino acids, it's energetically expensive. You do it with the help of ATP, which the power from the ATP hydrolysis, but not directly as ATP hydrolysis with water, is going to allow you to add this amino group where you had an oxygen here. So basically, you want to kick out this oxygen and replace it with a nitrogen in order to go from glutamate, which has a carboxylate group, to glutamine, which has the amide group. You can't just kick off the oxygen because that's a bad leaving group. Instead, what you're going to do is you're going to take the phosphate group, the phosphoryl group, so the phosphate with 3 oxygens, this phosphoryl group, add it to this glutamate. And then you're going to replace that with the amino group from ammonia. And so by adding that phosphoryl group, you are both making a better leaving group. So this phosphate group is going to be happy to be kicked off. It's got nice resonance stabilization when it's kicked off and things. And you're making it so that you've got more electron pulling away from this carbon. So you're making this a better electrophile so that the ammonia can come and attack this carbon and kick off this phosphate group. But the phosphate group was coming because you transferred the phosphoryl group from the ATP that left you with the ADP. So that's this part. And then when the ammonia comes in, it kicks off the inorganic phosphate, and that's where you get it here. And you're left now with your glutamine, in addition to the ADP and the inorganic phosphate. So although what you see written is this simple looking thing, what's actually happening is a little more complicated. But because of the state function nature of delta G, it has the same overall free energy change as if you were to just do this directly. If you need more energy, you get more energy going from ATP to AMP than you do from going from ATP to ADP. And so sometimes what you'll see is that things will get amylated. So you have AMP actually attached to a molecule rather than just a phosphate group. So things like where you hook the amino acid onto tRNA, that happens with this AMP. Furthermore, if you go from ATP to AMP, you also have this PPI, this pyrophosphate, which can then get hydrolyzed to give you another -4 kilocalories per mole. This is used to help power processes like DNA polymerization and DNA ligation. Although we typically talk about ATP as free floating, it's usually complex with magnesium cations, which stabilize it. So we're talking about metal catalysis as one of the ways that enzymes catalyze reactions, one of their catalytic strategies. That involved often things like magnesium ions helping coordinate with the phosphate groups of these phosphorylated molecules. As I mentioned before, other phosphorylated compounds, as well as things like thioesters, also have large negative free energies of hydrolysis, and some of these can actually be used to make ATP. Remember that we had a -7.3 kilocalories per mole to go from ATP to ADP, which would mean that if we want to just stick that phosphate group back on the ADP to give us ATP, we'd actually need 7.3 kilocalories per mole worth of energy. But if we think about the hydrolysis of phosphoenol pyruvate, now we've got -14.8 kilocalories per mole. So if we were to hydrolyze phosphoenol pyruvate, we get 14.8 kilocalories per mole, and we've got plenty of energy then in order to put that phosphate group back onto ADP to give us ATP. But we're not transferring the free phosphate group, we're transferring the phosphate group from the phosphoenolpyruvate onto ADP to give us ATP. When we do things like this, where we add a phosphate group or a phosphoryl group onto ADP to give us ATP from another organic molecule, we call this substrate level phosphorylation. As opposed to when we're adding it from an inorganic phosphate, such as with our oxidative phosphorylation reactions. A couple of examples of substrate level phosphorylation and glycolysis. So here we're transferring a phosphate group from 1,3-bisphosphoglycerate onto ADP to give us ATP. And here we're transferring a phosphate group from our phosphoenolpyruvate onto ADP to give us ATP. And note that the enzymes that catalyze both of those processes, we call them kinases. So kinases don't always transfer from ATP, but they're transferring from some organic phosphate source typically. Those steps are actually making ATP, but there are also steps of glycolysis where we're spending ATP, such as that first step that we talked about, the hexokinase step. And that hexokinase step is phosphorylating the glucose using the energy from the hydrolysis of ATP. Because free energy reactions are actually additive, so we can combine that glucose 6-phosphate, which had a positive delta G naught prime, and the hydrolysis of ATP, which had a negative delta G naught prime. We can add those together, and we end up with overall a -4 kilocalories per mole for the sum of those processes. So even though it didn't happen directly as two separate reactions, but rather these coupled reactions, the overall process, because it's a free energy change, this is a state function, so it only depends on the beginning and the end. We're able to add together these energies and get this thermodynamically favorable reaction. It's the sum of the two processes that makes it so that you're favorable. And the reason why you got so much energy from the ATP hydrolysis was not just that it was inherently favorable, but also that we were skewing the concentrations so that we keep the concentrations of ATP high. Other reactions of glycolysis that we represent with these reversible arrows, these are going to be metabolically reversible reactions, and so we can control which way things are going by controlling the concentrations. We can therefore use this pathway for both glycolysis, the breakdown of glucose, and gluconeogenesis, the making of glucose, except for these few steps that are metabolically irreversible, these single arrows, and those are the points where we're going to go around. And we're going to highly control those steps because they commit us to going in one direction.